So welcome everybody. There are still some people coming in, but it's great to have you here today. Happy to all participants on Zoom and our viewers on LinkedIn Live. A few basics beforehand, especially for Zoom, but also for LinkedIn, of course. Please mute your mics. Feel free to turn on your cameras. It's always nice to look each other in the face. Please post questions in the Zoom chat or in the LinkedIn event chat, of course, and we'll bring them into the Q&A session. Please feel free to participate actively in the Q&A. If you're on Zoom after the presentation, join our conversation. Please use the raise hand function on Zoom or just join in, and welcome to be here. My name is Aker Rudolph. I'm director of the UX Design Awards. It's my pleasure to welcome you to our deep dive. In these deep dive sessions, we present different award-winning teams, both from the professional sector and new talents, the projects and the stories twice a month. So just a look out in front. Our next one is a new talent track with a new talent award winner, Siddharth Ahuja, and his app Yo Guide. It helps visually impaired people to exercise independently at home. It'll come up in exactly 2 weeks, 30th of June. So please check our website, stay tuned for this. But of course, why are we here today? Our project today that we present is a Gold Award winner in the UX Design Awards. So I'd like to say a few words about the awards. It is a global competition for excellent experiences. It is organized by the International Design Center Berlin, IDZ, a leading German nonprofit and member-based institution based in Germany. Our mission is to promote the value of design in business and society. And by doing so, we founded the awards back in 2015 to raise the recognition and the value of positive experiences. And of course, both in the professional sector, in services, products, prototypes, in visionary concepts, but also in the works of young designers. So if you've got great experience projects in the works, feel free to have a look at our website. The next call for entries will start in September. But the competition is not only about promotion. We created the awards because we believe that good UX design has a huge and ever-growing impact on shaping a positive living environment for us all. And this is especially visible in the healthcare field, where every life counts. And the past year, I think, has shown us really how important healthcare, diagnostics, which we'll be talking about today, and a well-equipped healthcare system are. So good experience design helps us to create solutions that are relevant, improve our lives, help us to achieve our goals in a better way, are easy, understandable, and hopefully even fun to use. And also good experience design can empower us, improve our way of life, our abilities, open access to knowledge, which we'll be also talking about today, and by doing so, strengthen inclusion in society. And this is ultimately why we created the awards also as a platform to promote great examples of positive experiences across all industries and fields, and to create a community of professionals and a platform for knowledge sharing and networking in UX. And this is why we're here today. So without further ado, I'm happy to welcome our guest. It's my pleasure to introduce Henning Falk. He's responsible for life sciences and imaging technologies at Intuity Media Lab. This is an innovation and design studio based in Stuttgart in the south of Germany. And he's the person leading the team behind the Gold Award winner, Minimic, the intelligent microscope. The jury of the awards awarded the gold title to Minimic for its great relevance. It applies state-of-the-art technology to diagnose infectious diseases that threaten millions of people around the world. The device is compact and features high-quality workmanship. The software is intuitive and easy to learn, and this relieves medical personnel and also increases the acceptance as well as the usefulness of the product, both in emergency situations and in remote regions of the world. The machine learning algorithm that is used here is open to further development by the community. And all summed up, Minimic is a positive example of how technology and creativity can be successfully combined. So this is not my words, these are the words of the jury. And with this great introduction. I would like to introduce our speaker, Henning. He combines a background in molecular biotech with a passion for tinkering with technology and creative problem solving. At Intuity Media Lab, he applies his skills in software and hardware prototyping. He tests and develops innovative concepts for a smart future. So it's a pleasure to have you with us, Henning. And I'd like to give over to you now.
Okay, thank you very much. It's great to be here. I think it's an awesome new format. I'm excited to share our experiences with the Minimic from before the award and also now afterwards. So we won the Gold Award in 2019 with the Minimic. That was fortunately still before Corona, so we had an awesome evening at the IFA in Berlin. And now, of course, it's home office time, so I'm in home office. But I think though there are a lot of disadvantages of this kind of situation, it's also quite good for formats like this. I don't think all these Zoom possibilities, all these meetings that are now so easily possible would have been like that in different times. So I'm quite happy that such a format now exists and that we can share a little bit of our development in the field of the Minimic now. Yeah, let me start with my presentation. Now it's sharing time. Okay, so I hope you can all see my screen now. Before I really dive into the topic of the Minimic, I would also lose a couple of words about Intuity in general, because I think it's a quite unique environment, also like very much influencing the way how this project, how the Minimic evolved in the first place. So the favorite way for me to explain what and how Intuity works is actually to show who Intuity is, because Intuity is really just the people that are working there. We're a young and dynamic team of about 30 motivated and excellent people. And we have this common goal that, to change something, not the entire world, that's maybe a little bit too much, but something about the world, like make it more efficient, make it more reliable, more connected, or just simply more fun. And with our very diverse expertises at Intuity, I think we are very well equipped for that task to shape meaningful products. We have people from all different kinds of backgrounds. Me, for instance, as a molecular biologist, mathematicians, architects, user experience designers, software developers, and that way we can offer our full spectrum of services in the hardware, software, UX, and strategy worlds, really. And usually what Intuity does is service development, but we also have our own projects. And the first and the longest and so far most successful product is the Minimic. This is 100% developed at Intuity from the first idea. And I think it wouldn't have been possible to go that far with such an also, yeah, new idea and difficult start also in a different environment where we wouldn't have these many different people working together and bringing in their expertise. Now the Minimic idea really started quite abstract as the idea how to exploit deep neural networks, trainable algorithms, AI, to transfer knowledge and expertise from experts to a larger community via trainable algorithms. This, in the first place, didn't have anything to do with diagnostics. The concept didn't even involve microscopy. But our view on the chances of deep learning really was that the possibility to transfer implicit knowledge to an algorithm, and therefore from expert doing a specific thing a lot of times to a broader community. Because in traditional data analysis and classical machine learning, usually you have to formulate explicit rules for the algorithm in the end to be able to classify certain features. Like example from my hobbies, I do bird watching in my free time, and a lot of times there you don't see the birds, so you have to listen to their songs. So obviously you train to recognize birds by songs, but if you look at all these guidebooks, the description how a specific bird species sings is very abstract. It's long words with a lot of vowels nobody can pronounce, and it's enriched with words like it's melancholic, it's sad, or very loud and happy. These are all descriptions that don't really help you to recognize the robin, for instance, in nature. The only thing that helps is having examples, going out with an expert, or watching videos on YouTube, listening to CDs. So we as humans are very capable of learning the implicit rules of a complex problem just from examples. And this is the big hope also for deep neural networks, that they can do the same, that just by feeding a lot of well annotated data, they are able to learn these implicit rules all by themselves. And of course, that's the shiny version of how deep neural networks will solve all our problems. Of course, anybody who has experience in this field also knows that it's not always that easy. And I'll also talk about the difficulties that we have on the way a little bit further on. But now coming back to the Minimic. So we wanted to create a practical example of this concept of AI enabling the community with the knowledge of experts. And biomedical imaging is really rich, really full with a lot of these perfect problems, actually, because there's a lot of image analysis, which is done by experienced experts, yet these tasks are extremely repetitive and extremely boring also. So it would be very convenient to somehow help and automate these tasks. And the use case that we're currently focusing on is tuberculosis diagnostics. When I presented the Minimic at the UX Design Award in 2019, we were mostly talking about our first pilot use case, that was malaria diagnostics. Now we decided to focus for bringing the device to the market on tuberculosis, which is the second leading cause of death from an infectious disease after HIV, with 9 million new cases of active TB every year. And it's not only a third world problem or a problem of emerging countries, but it's also still quite a problem in industrialized countries. So for instance, here in Stuttgart, we have a partner lab, and they do analysis of tuberculosis samples. They have about 30 to 50 samples per day. Most of them, of course, are negative, but still it shows that this is not just something that happens somewhere else on the world. Now, what we see here on the screen is a stained sputum sample. So it's basically, if you cough and then get a salivary sample, this is what is presented here on a little glass slide. The glass slides look basically like this here. And the sample is stained so that the background is bluish, and these little red rods are the tuberculosis bacteria that people are looking for when they diagnose tuberculosis. Now, this is a very easy sample because contrast is good, and we have plenty of these little rods in the one field of view. Now, in reality, especially if you have very mild tuberculosis at the beginning, you have to look at over 100 of these fields of view, and maybe you find something between 5 or 10 of these rods. So there's constant, very high concentration needed, and most of the time you're looking at blue background with nothing important happening. Now, because microscopy is very cheap and relatively fast, and microscopes are available all over the world, it's very widely spread as the #1 diagnostic tool for tuberculosis. Yet, and that really surprised me and shocked me when I looked at numbers, it's extremely unreliable. So only 60% of the patients that were diagnosed using microscopy are actually correctly identified as tuberculosis positive. That's a very rough estimation, and I think it really greatly varies between labs and countries. For instance, in India, there's a big lack of trained personnel that can properly actually identify tuberculosis in samples. So on top of the relatively poor coverage of medical service in rural areas in India comes this lack of good diagnostics, diagnostic quality, which makes the whole problem even more pronounced. Now, this is where we wanted to start with the Minimic. We wanted to build an alternative to reduce the error rate and to help the diagnosing person by sorting out most of the negative cases and the clearly positive cases, only leaving the few that are really borderline for a trained person to look at. Because what we also very early learned, and also if you look at other solutions that are already on the market, usually they are all, especially if it's about image analysis, they are assisting systems. So the final decision how to treat the person always lies with the doctor. These systems always try to augment samples, point towards specific features to help the person make the judgment, but in the end, decisions are done by the medical expert, and this is also where we are aiming at with the Minimic. I think for us, very key was from the beginning that we didn't want to have a software alone, but to combine hardware and software in one solution. Because, well, on the one hand, of course, it would be very convenient to only have software, because then everybody who has already a microscope could use that just on top. Yet it makes the imaging or image analysis problem extremely difficult, because each microscope has a little bit different optical properties, cameras are different, and therefore the complexity of the problem increases the more imaging modalities you really allow in the system. And to be able to do diagnostics or to help diagnostics, you have to ensure a high quality, and this is much easier and much more reliable if you have a close fit between the hardware you're using and the software for the analysis. Now, for those that haven't seen the teaser video, I show a little snippet from our Minimic video, just to give you a little bit of an idea how it looks like and how it moves. So we have an upright microscope with an objective here in the middle and a table where you can put in the glass slide with the sample. And this is our user interface. It's browser-based, so you can look at it at different devices. It's extremely simple. So basically, you only register how many images you want to take in the sample. It gives you a direct feedback which images of the stack of images may contain parasites or bacteria, and then you can look through the critical ones to confirm that the algorithm was right or wrong. Now here what you see are blood cells. This is still from our malaria prototype, and now we are working on tuberculosis, as I said. I actually also have my Minimic right here with me. I just hold it in this little window up here. Just for you to get an idea, this is approximately the size. It's basically a coffee machine. And tomorrow I'll go to a lab in Heidelberg to capture images. That's why it's now standing next to me in home office, waiting for its great day tomorrow. Okay, so back to the concept. Now putting together software and hardware in one product is a bit of a challenge from a market access perspective, because usually systems that include optical hardware are particularly expensive. And as I said in the beginning, where it's mostly needed, these are countries where expensive devices are very difficult to sell. So we realized really from the beginning that it is crucial for the Minimic concept to have a very cheap hardware combined with an intelligent software to somehow hit the sweet spot between the currently installed manual microscopy on the one side and the fully automated systems that do exist, yet are only practical for very large labs where there's a high throughput. And we wanted to aim into this middle sector of small and mid-sized labs. And I think when we designed the Minimic, we had actually an advantage not to be one of the classical microscopy companies, because we approached the topic kind of really bottom up without any technical legacy. We tried just to be good enough and see what's happening. And I'll show you a little bit more in the next slides. I think I already mentioned in the beginning, Minimic's 100% Intuity. It's really cool that kind of Intuity gives this possibility to develop a product from the idea, first prototypes, hardware and software, to the first mini series, which here Marcus, our CEO on the left side, and Albert, a retired lung surgeon and a friend of Minimic, are putting together here. So that's our assembly line. Albert was the one that came up with the tuberculosis use case in the first place and established for us the first contacts to labs. And here he's also helping building the first 10 Minimics that are now working in different labs to collect data. Yeah. So let me talk a little bit about how we approach this topic and why our Minimic on the one side looks a lot different than the classical microscopes that you're used from Leica or Zeiss or Olympus. So traditionally, the microscopy industry strongly focuses on precision, optical quality, and also on versatility of the use cases so that you can use one microscope for a lot of different applications. So it's a very engineering-centered discipline, really. Now, for the Minimic, we try to be a lot more agnostic on the technology and really focus on this one workflow that we want to tackle, the diagnostic workflow. And by that, we could kind of eliminate a lot of the very complex parts of the classical microscope. That's why on the one side, you have all these no's. These are all the decisions we took, what we don't need, or where we could go a lot more easy or cheaper than what the classical microscopes would include. A couple of examples, for instance, we don't need the oculars to look through to actually see the sample because everything is digital in our microscope. It's a digital only microscope. That makes it also a lot easier to make it small and compact because you don't have to adapt to the body to be able to manipulate it. We have very cheap and simple optics. So our first objective that we used cost 25 euros, while on a high-class microscope, these can be easily a couple of €1,000. Of course, I talked about the AI-based analysis. We have a digital autofocus that doesn't need precision stages, but just takes the most focused images from a video flow. So it's also very vibration tolerant because we have this very high capturing rate of images. So you can even carry the Minimic while it captures images of your sample. I don't know actually why you should do that, but it's possible. And of course, browser biased user interface, you don't have to buy this big machine for the analysis coming with a microscope. Yeah, actually recently I read a scientific article describing the tendency of people to optimize systems by rather adding more stuff to them than by subtracting things. But I think here with the Minimic, we really tried always to first take things away and test whether it makes it better or not. I have to say that a couple of times we also had to add in things back. So we started, as I said, with a very simple objective, which didn't really function with the field of view that we needed. So we stepped up a couple of steps. Also, we started with a simple webcam in the back of the microscope. Now we're stepped up a couple of steps in the sensitivity level of our camera to be able to capture the images at the rate that we need. But we always try to just go as good as you need to have a digital microscope that can analyze images by an AI and not by a human being, because also there in the image quality, there's a lot of, let's say, room to play if not people have to look at it all the time, but just an algorithm. Now that was about hardware. Let me also talk a little bit about software. So the initial concept was very much community-centered. So our idea was that people, experts, image samples with a Minimic, and then with an integrated tool in our software, they can annotate these images, which means they mark the features that should be recognized. And then these images are loaded into our cloud, and the trainable algorithm goes over them and generates a new network specifically for this use case. And this then is shared in the community. Thereby, you have this very bottom-up possibility of transfer knowledge from experts that dedicate their time and their expertise to a specific topic, and then the entire community can benefit from that. Now, still, I really like this concept a lot, and I'm pretty sure we'll come back to that at one point. But right now, we're taking a different route because from the beginning, we wanted the Minimic to have a diagnostic value. But if it is used in diagnostics, it has to be somehow certified, CE approval or an FDA approval. And this system involves obviously certain standards. The analysis has to be extremely reliable, reproducible to be able to be certified. And this bottom-up community approach there doesn't really work because from one day to another day, with the input of new data, the analysis results could vary, and this therefore could not be certified. That's why now we take a little bit of a different step for the tuberculosis use case. That's why I have the Minimic with me for going to the lab tomorrow. So we're collecting together with different TB labs in Germany sample images from tuberculosis samples, validate, annotate them, to put together a validated, certified algorithm that can be sold together with a Minimic as a specific dedicated tuberculosis Minimic solution. Now, these are a few images just to show you where we are with the analysis. So that's these are sputum samples acquired with the Minimic, and the red boxes are all the tuberculosis bacteria that the algorithm found. And so far, it actually works quite well. You see even down in the bottom of the image, these unfocused bacteria are pretty well recognized. And also this big hair swimming through the immersion oil doesn't confuse the algorithm too much. However, the big challenge with deep learning is that always the first examples look pretty good and you get very fast and far in the beginning, but the further you open up the variability of the images and the input, the more challenging it becomes to hold this high standard of sensitivity and specificity. So if we want the Minimic to work on all different kinds of samples, and TB samples can look very different, with also red parts in the back being lighter or darker, then this is quite a difficult topic. And I think most or a lot of the problems why there's so much heterogeneity in the images or in the samples, in the prepared samples, is that the entire workflow, the manual workflow, is split into a lot of different steps. So from sample collection to smearing the sample, fixing and staining, to the microscopy. These are all manual steps. For each, there are different protocols. They vary between labs, and therefore, in some, you get very heterogeneous results for microscopy. And that's, I think, also part of the reason why the smear microscopy performs so poorly in terms of actually recognizing tuberculosis. Now, of course, there are two basic ways how to approach such a problem with variability. The first one is to make the analysis more complex, throw in more training data so that you can capture the entire, hopefully the entire spectrum of possible samples out there, and make the analysis pipeline more complex. For instance, dividing samples into different types, light background, dark background, more reddish, more blue, and then train separate algorithms for separate parts of the problem, really. And the second option is to fight the variability in the first place, trying to get more homogeneous input. And as usual, if you really want something, you don't try with the first thing and then go to the second, you do everything at once. And that's also what we did. So we are still working on optimizing our on our training platform, now separating the problem, the analysis problem into subtasks that are then fed to different neural networks. And on the other hand, we're also looking at this whole process of this whole workflow of tuberculosis diagnostics. And there, on the one hand, of course, we have the Minimic that unifies the process of imaging and the interpretation of the images. So that gets more unified. But we also wanted to go further towards the beginning of the journey. And there we are now partnering with a company that produces equipment for the automated sample collection and slide preparation and staining. So that now together, I think that's really an exciting match that we found there. We can cover the entire workflow from sample to result. And that's really exciting because that was for a long time already our slogan, intelligent microscopy from sample to result. And with this cooperation, that really becomes reality. And still, such a semi-automated workflow is a lot more flexible, and the equipment is a lot cheaper than the fully automated robot labs that are already on the market. And of course, beyond the obvious benefit of such a seamless workflow for the user, it helps us a lot with the image analysis because it releases the pressure on being able to recognize any samples that are out there, and we narrow down the variabilities a lot. I think this is my last slide, future beyond tuberculosis. I think the cool thing about the Minimic is that it is a platform. Both the hardware is very generic as well as the pipeline for generating new analysis algorithms. So it can be adapted for a lot of different other analysis problems in medicine, in life science research, but also in other disciplines. We were talking also about material size, quality management, food and drug discovery. And it's really exciting always to talk to people from this different areas, from different disciplines, because whenever I show the Minimic, usually somebody comes up with another idea where such an automated system could actually be beneficial. So I'm really much hope that tuberculosis is our first successful application of the Minimic that we can see also on the market. But I also am quite excited to see what else might come from this generic platform. And I very much also hope that we get back and see this more community-based model, maybe in a more scientific research device. Yeah, that's an exciting future, and I'm much looking forward to see the further development of the project. Okay, with that,
wonderful.
Stop sharing, I guess.
Thank you very much, Henning. That was a wonderful insight. And to all of you out there, please, as I said again, join the conversation. Raise your hand, write in the chat, or also write through the LinkedIn chat so we can take up your questions. But Henning, the one thing you didn't talk about, it's a bottom-up project. It's a wonderful big effort for our design agency to be involved for years in this development. But where did the idea come from? What was the spark at the beginning?
But I mean, I think that's that's part of the culture at Intuity. We like to think about what the future might bring, and our way of seeing the world is always trying to use the current technologies that are around and thinking up new ways of combining them and making a better use and the more valuable products from them. And of course, great if that happens in our service business, but you know, getting ideas and thinking about concepts doesn't stop with that. And here it was just that the concept just looked so tempting to try it somehow that we really just had to build the first prototype. And from there, actually, first it really kind of evolves by itself. So can we do imaging? Yes, we can. First trained network also already looked quite promising. And from there, it shaped to a more and more complex concept that now already, evolved since 2017 at Intuity. Yeah. I think you're.
Of course, I think it's very important that, or maybe it's a question to you, that you come from a medical field, because I understand that you found a niche, you found a problem in a very specific field, and you decided to tackle it, right? It doesn't seem to me something that anybody would come up with.
Yeah, I think that's also, again, the chance of being with so many experts from different fields together in one, in one lab. The idea, as I said, for tuberculosis came from Albert, our retired surgeon, but of course now kind of the understanding of what a lab really needs, like a medical lab. This is also a lot of my experience from the times back when I was still myself pipetting. But I mean, that's only one part. Then the whole understanding of how much we can actually take advantage of the whole 3D printing environment, all the boards, the drivers, the motors, how much you can actually benefit from this very rapid development to build something small and cheap that goes into the direction of microscopy, where these concepts aren't really, it's all still glass and brass and very high quality. A lot of ideas come together to funnel into such a complex product like the Minimic.
So what would you say, what's the true game changer in Minimic? So where does it really score? And maybe also following up to this, what are the fields where maybe it doesn't have, so many advances compared to the status quo?
Yeah, I think the big game changer, the USP, is really the simplicity of the usage combined with this extremely robust, low-cost hardware. Because any of these solutions that are out there, especially in the medical field and the science field, that somehow try to automate these processes, just go very quickly into the hundreds of thousands euro, because there's a certain legacy in how these problems are tackled. And I think that's really the big advantage, that we have this platform where we very quickly can test new ideas, new use cases. We didn't start with tuberculosis, but now found out that this one is a very, very promising one. And this is really where such a system can count. I also said, like in the bigger labs where automation is on a much more sophisticated level already, the Minimic wouldn't be ideal, because we also have to make compromises with speed, for instance. Since we don't have very accurate positioning, we need more time to find the focus at each position, for instance. Now, there are different other solutions that are much more efficient. But again, it comes at a certain cost. And it's surprising how many labs, especially in Germany, it's very diverse, how many small labs do still these kind of analyses with 30 to 50 samples per day. And their fully automated robot labs would be just total overkill. So it's really, I think, again, coming back to this sweet spot, where is it too much work? to do it by hand all the time, and when is the point when it's worth investing into something more complex and fully automated.
So what would you say are your clients and application fields then? Is it a bigger doctor's office, several doctors? Is it a developing country? Is it an emergency situation? Mm-hmm. Is it a remote place?
So right now with the Minimic in its current configuration for tuberculosis, it will be the diagnostic labs. So each practitioner, medical staff send in samples, and they are usually diagnosed in a lab. So this whole staining process is still relatively cumbersome. So people tend not to do it directly in the doctor's office, but send in the samples. And this is where I would see the Minimic first. We're still trying to understand the concept or the way how this chain works in India and in China mostly, because there's also, since you don't have this several layers of first the doctor sends in the sample to the lab, maybe the lab actually sends it to another lab because they have enough samples, pool them, and everything comes back. This whole structure isn't that developed there. So there you have more also these local TB stations where you can get direct diagnostics. And this is the second part where I see our microscope very strongly, directly basically at the patient side in countries, in areas where the tuberculosis incidence is really high. We were also thinking about a version with a battery pack so that you can actually, kind of drop it in the jungle. Not sure whether this is really the first thing we'll try. Yeah, but I think in doctors' labs and in the labs, that's where it's going to be useful.
That obviously means very diverse user base. So very different users on a different level, and you've been talking about India, China, so completely different cultures, work processes. So what would you say is the biggest challenge in terms of user experience there? Because a part of the solution is an intuitive software, and that really helps you. So how are you working on this, and what are maybe the biggest challenge you would say in terms of UX design at the moment?
Yeah, as you pointed out, the highly diverse crowd that is going to use the system is a challenge. Of course, language, also the way how to interact with these devices. Yet for the Minimic, I think we're on a very good level because the usage is extremely simple. Basically, it's very much oriented at the consumer level, user interfaces. So it's a little bit, yeah, it guides you through the workflow quite easily with selecting which samples you have, click OK basically, and then the system does the rest of the job. Where it really gets kind of complex is the second step of fusing the knowledge of the algorithm with the experience of the doctor, of the medical person. Because as I said, we don't give a yes or no answer. We say most likely it's negative, it's positive. Look please at these couple of images to make up your own mind. So we point out certain regions of the sample where there might be tuberculosis. And this is now, of course, very crucial on the one hand to inform the user about enough of these regions so that nothing is lost, but on the other hand, not overwhelming the user with a lot of sample regions where there's possibly something, because at that point, suddenly you don't have a benefit anymore because you're looking at images instead of the sample in the first place. It doesn't really help and doesn't get it much faster. And you get, again, distracted and tired, and the whole quality of the analysis suffers. So it's really this fine balance between being sensitive enough not to lose anything, but on the other hand, not overwhelming the user with too many possibilities and with too many maybes, so that it actually increases the throughput and helps the medical person.
So this is a fine balance between the collaboration between human and AI, right? That you need to
Exactly.
thread. It's a thin line. So how do you come at finding the right balance?
Now we talk a lot with the people in the lab, how the process of building trust to such a system would work. Obviously, people working with microscope for a long time have a very specific view on how a sample should be represented, how the colors should be, how much of the sample should be visible, how to manipulate the focus to be able to judge the samples. And this we have to reconstruct also on the Minimic to be able to kind of give the user the confidence that he or she can judge upon the images taken by the device. For instance, setting the color scheme in the way that people would like to have it in the lab. We changed a lot of LED illumination sources until we got something that had enough red content so that the little bacteria really stood out as very bright red. We thought like purple is good enough. Apparently it wasn't, it has to be red. And then on the other hand, it's the question of how many samples do people want to see before they are certain to make a specific decision. And that's also something that we discuss a lot. How much would you want to see? Which samples do you want to reprocess? Do you want to reprocess the entire sample or only look at the images? So we are very much in contact with the people in the labs. And this is an advantage of that we anyways have to go to the labs and collect a lot of data, that we are very close with them, and they give very direct feedback on what they would like to see and when they would think it's a valuable addition to their workflows.
And do you offer any personalization? Because that sounds like, well, if I'm a doctor in Germany, maybe I want more red and somebody in another country wants more blue because the hue of the material they used to is different. Mm-hmm. So is that any kind of topic in this kind of field?
Yeah. So basically we have two levels of the image. So there's the raw format, which is fed into the algorithm, because the algorithm always wants to have the same input data. But then we have the second level of adjusting hue, color balance to the needs of the user, which isn't actually saved in the raw data, but is only on the display. And that way you can at least buffer some of the specialties and the special wishes of the medical stuff. Yeah, to accommodate exactly that.
Mm-hmm. And obviously there is like a business question to it too. And if somebody wants to invest in the machine, so how much better is it when you train on this, on Minimic compared to other systems? Is there like a steeper learning curve? How much less time does it take?
I mean, if you actually use the Minimic, the one thing that is much better is that you don't spend, or the technicians in the lab don't spend this long time looking through oculars into the microscope. That's very exhausting. You get a stiff neck and and also deteriorates eyesight. So that's a very exhausting work. And it gets increasingly hard to find people that are willing to do that kind of job. So it's an incentive for labs to have systems that work more on a office desk kind of workspace, where you can spend more time without wearing away the people that are working there. That's the one bit. And on the other side, you have a lot less hands-on time on the Minimic, because usually on the microscope, you spend 15 to 20 minutes looking at different positions, writing down the findings that you made. With the Minimic, it does all the imaging at the first place in about 5 to 10 minutes, and then you get the summary of a few areas of the sample that you have to look at again. So the hands-on time, really interacting with one sample, goes down from 15 minutes to maybe 1 or 2 minutes, and that's one of the big advantages here.
Right. So you take the KPIs already on that. So you mentioned this. Your looking for partners, and you're already quite ahead in maybe your quest for partners. So what are the next steps? Because it's a very complex market. Now the hard work starts, right? When it comes to safety regulations.
Exactly. Yeah, ideas are usually cheap. Then it gets more increasingly difficult once you want to bring them onto the road. Now we are in the good situation that we are very close to closing a deal with a company to bring this device onto the market, because we don't have the access to markets. We don't have a sales force going from lab to lab and presenting the device. We don't have experience with FDA approval, CE approval procedures. And that's where partnering gets very important. And this is exactly where we are right now. So figuring out who does what, who develops what, where do we produce the device, and how much actually do we need as input data to do a validation study to get the approval for an FDA and a CE mark.
So it's maybe a kind of comparison, such as Pfizer and the German company that developed the COVID vaccine, that you have a bigger brother who knows the industry and can help you to actually understand the system and make it workable on a scale, right?
Exactly.
So if you see yourself in 5 years, where will you be with Minimic?
Well, I very much hope that in 5 years we have it on the market already, something like 2 to 3 years. So development time right now is estimated to be another 2 years. And then we hope to be successful with the tuberculosis, both in the Western world as well as in the developing countries Far East. The exciting bit also about this cooperation is that they also offer automation solutions for other topics. So beyond tuberculosis, again, going back to malaria, other disease diagnosis. And of course, if we build on a platform that is already established in the market, it's much easier to bring in the next version, the next use case. And so we are hoping that we can fill the pipeline for the next couple of years, bringing in new valuable solutions to complement the staining and preparation equipment that is already out there.
And maybe a final question, because this sounds like you're going into the market funnel, which means, of course, you have to shape it and make it fit the market and also the expectations there. But you mentioned platform very often, and I think the open source baseline or the original idea was fascinating, that basically the medical community could train the AI to different scenarios, different use cases, different diseases, and contribute to a knowledge network and a platform. So how would you hope to establish this? I think you said a few words about this in your presentation, but does it exclude the market approach, or is it something that can be built into it or even enhance it? Any ideas on that?
Yeah, I think there's a good possibility to, once we know how the market works and we have a certain reputation that this system is actually valuable, you always have these two lines. The one is the diagnostic medical market. There's only very little flexibility, because obviously it's a decision on the health of people. But many of these labs also have research brands, or like universities with hospitals connected to that. So you have this basic research and applied research branches that are always looking for testing new systems, developing new systems, and that's perfect for this community approach. When you're trying either to optimize your mouse model on understanding a specific disease better, and you have to count certain level of for instance, malaria infected cells in the system. This would be extremely helpful if you have a system that you can train specifically for your use case, and then kind of on the second level, then people benefit from that because research gets more efficient. And this is, I think, where this teaching community-based approach is very powerful, because usually these are very specific problems. Not a lot of people use the same mouse model with the same kind of image analysis question, but there is a certain very active community usually, and in such a community, these algorithms could be developed, and this is then again inspirational source for also us to understand what's possible with the system and where we could also branch off another dedicated system commercially available then also for diagnostic labs. So I think as a ideation platform, let's put it like that, it's very, very valuable. It's going to be the second step, not the first step. First, we try the hardware with tuberculosis.
Wonderful. Last question. I don't get any from the crowd, so I think that we're really good in putting the right questions on the forefront. But oh yeah, Wolfgang, please just join in.
Hi, thanks, Henning. Great product. I'm a big fan. It's really super cool in this area to reduce so much complexity in the field of light microscopes. Great. I'm really a big fan, and I'm UX designer from Munich, and I was working on SandBlue for Zeiss for 3 years. And that's why I'm a little bit into in this field. And I have the question: is there the possibility to check out the device somewhere? And I would be super curious about your UI, because what I could see in the videos looks really super cool. But I would be very interested to see how you solve some detailed questions, how your workflow looks like, and is there the possibility to check it out and to play a little bit with the device sometime?
Yeah, I mean, let's say in non-Corona times, we always are very happy to invite people just over to our office at the Feuersee in Stuttgart. So that will be a perfect opportunity, and hopefully, fingers crossed, the fourth wave doesn't crash over us too soon. So come by, visit us, and you'll get a full preview of the current status of the Minimic.
Yeah, I would be super curious and super interested. Thanks.
Well, the next step would be just hook up over LinkedIn and then make the next steps. Wonderful. And also you mentioned that you're looking for talent both in software and AI development. So anybody wanting to work with such a great team, highly welcome. I don't get any more questions. I very much hope, or from our side, we very much hope from the Design Center that also winning the award helped you in your development and your story, and also in such a, let's say, very specific medical field that is maybe not closely associated with user experience. That's changing very fast. I hope that was useful, and as we see from many submissions in the last years, I think the medical field is extremely dynamic, and it's completely going out from the niche and the specialists towards really a take up, as Wolfgang said, of user experience and really an approach to people, as you wonderfully explained, Henning. Thank you very much for this. Thank you very much for these insights, and good luck for the next steps in Minimic. We're holding our fingers crossed and hoping for the best, and looking forward to see where you are in 2 years, and then we'll hook up again about the next 2 years. So thanks for everybody. Thanks for being with us. And of course, thanks to our team, Magda, Fabian, Billy, and Jake. And I would like to remind you just quickly, it would be wonderful to welcome you again to our next event, exactly 2 weeks in June 30th, with the UX designer Siddharth from New Delhi in India. He developed an assistive tech solution for visually impaired people, and he is now founding his design agency in India after being with the Copenhagen Interactive Design Lab. So please check out our awards website for more, and the registration for the next event, of course, on the awards. And very soon you will also have the recording of this session, so you can reread it, rehear it again, re-see it again. And I very much hope to see you again and to be in touch. So hook up with us via LinkedIn, all our other channels. And I hope that we'll be able to welcome you again. Thanks very much, Henning, again. Thanks to everyone, and have a nice evening, have a nice morning, wherever you are, and see you soon again. Bye-bye.